Just to review before we go forward what all this is based on. Hopefully you're aware of this already, but meiosis really explains why we're getting the results we do. So here are two parents. They were heterozygous. So this parent might have been big G, little g, so heterozygous. This parent here is big G, but you could imagine little r, little r. Through meiosis, they produce 4 haploid gametes, and they're only going to pass on one of the 2 homologous chromosomes. So in the possible gametes that this parent produces, 2 of them have a big G, 2 of them have a little g. In the possible gametes that this parent produces through meiosis, all of them have a big G. So then when you have fertilization, randomly one of these gametes will meet up with one of these gametes. There's a 100% chance that you're going to get a big G from this parent because all 4 of them are big G. Just like there was a 100% chance that this parent here from that first question today gave a little r. And this parent has a 50% chance of a big G, 50% chance of a little g. So the possible offsprings from this cross were big G, little g, or big G, and it'd be 25 or 50 and 50% for that. Just like here you had big R, little r versus little r, little r. So meiosis, just so that you connect this, so that you really understand why this is, meiosis explains why this happens, that this parent can either give this gamete or this gamete. This parent can either give this gamete or this gamete. So these are representing gametes. And this is going to be important today when we start looking at dihybrid crosses, because the gametes are going to get a little more complex on how we figure them out. All right, I've got one more question before we go into dihybrids. This one's a little bit longer, so I'll read part of it to you. Imagine you're a genetics counselor, and you're working with a couple who have just had a child who is suffering from Tay-Sachs disease. Neither of the parents have the disease, so neither parent is positive for Tay-Sachs, nor does anyone in their family, so none of their relatives have it as well. Which of the following statements could you, as the genetics counselor who knows all about genetics, make to the couple? A, because no one in either of your families has Tay-Sachs, you are not likely to have another child with Tay-Sachs. B, because you have had one child with Tay-Sachs, you must each carry the allele. Any child you have has a 50% chance of having the disease. C, because you have had one child with Tay-Sachs, you must each carry the allele. Any child you have has 25% chance. D, because you have had one child with Tay-Sachs, you each carry the allele. However, since the chance of having an affected child is 25%, you may safely have 3 more children without worrying about another child with Tay-Sachs. Or E, you must both be tested to see who is a carrier. So which of these is something you could tell these parents? Okay, you guys are doing pretty well with this. It's good. I'm going to stop this. So Tay-Sachs, is it dominantly inherited or is it recessively inherited? All right, how do you know recessive? Okay, both parents don't have it, they have a child with it. So you're already thinking ahead. We're going to talk about using reasoning like that in the future to make something called a pedigree. But you're exactly right. The only way that 2 unaffected parents can have an affected child is if it's a recessive trait, as they have to both be able to be carriers of it and not express it. So Tay-Sachs is recessive. So I'm going to use a little t equal negative, and a big T equals, so positive, and a little t equals positive, and big T equals negative. Right? Okay, so both of the parents are what then?
Carriers, but they're hetero.
Okay, so they have to both be big T, little t. If one of them was big T, could they have a Tay-Sachs child? No. And you can do this cross. So you've got big T, little t from one parent, only big T from the other parent, and you end up with big T, or big T, little t, neither of which would be positive. Right? So the only way to have a Tay-Sachs child would be for them both to be big T, little t, in which case you'd have a 1 out of how many? So 1 out of 4 chance of having that child. So do we need to test them to see who's a carrier of Tay-Sachs? No. You know both of them are, right? As long as they're both the parents, you know both of them are carriers of Tay-Sachs, right? So you laugh, but that's actually something genetics counselors have to worry about. They sometimes do have to do testing to make sure that the parents are the actual parents, right? So what if they have another child, what is the chance of them having an offspring with Tay-Sachs? Right, 1 out of 4, right? 1 out of 4. So it's 1 out of 4. So we can really, A is not right because they still have a 25% chance of having another child. B is not right because it's 25, not 50. E is not right because we know what they are. What's wrong with D? None of you chose that.
Because it's a possibility that they'll have 3 other children.
All right, so.
You don't just take away that percentage.
Okay, yeah, so it's not, so this would be, if D was true, this would be equivalent to I flip a coin, I get heads, and so now the next time I flip it, I'm going to get tails, right? So that'd be really easy for football players. They would just flip the coin ahead of time and then know, okay, the next time it's flipped, I know it's going to be the opposite of that, right? So it doesn't work that way. Every time you flip a coin, it could be heads or tails. The percentage comes to play when you do it over and over and over and over again. So if these parents had 100 children, chances are 25 of them around would be Tay-Sachs positive. Probably more like 23 or 28, something around that area. But the more and more children they have, the closer it is to 25%. But they could have 2 children back to back with Tay-Sachs. Okay, good. Most of you chose that. Those of you that didn't choose the 25%, probably said needs testing. But can you see why you don't need testing? Because you know that the only way to have a child and have 2 parents that are unaffected is for them both to be carriers. So good. So the answer was C. Okay. So I want to kind of blank this for now. Okay, so I want to just do this on the board because it helps if we write this out a little bit. So we've been talking about what are called monohybrid crosses. So one trait being passed from generation to generation. That's not all that interesting genetically. We don't really care about there being, just looking at one trait at a time. We usually want to know a whole bunch of different traits at the same time. So, okay, so yellow, just to set this up, yellow is going to be dominant to green. I'm going to give yellow a big A, green a little a, and round is dominant to wrinkled. Okay, I'm going to give this a big R and a little r. So I want to do a cross between a heterozygous yellow and heterozygous round pea plant with another heterozygous yellow, heterozygous round pea plant. So now instead of just having one trait, it's either yellow or green, I now have 2 traits. It's going to be yellow and it's going to be round. So this would be big A, big R, little r, crossed with big A, big R, little r. Okay, so those are my parents. This is my parental generation. To do this, the first thing I need to do, just like I had to in the monohybrid crosses, is I need to figure out what gametes this individual is going to produce. So to do this, I need to consider where these lie on chromosomes. And because we're not talking about gene linkage yet, which we'll get to next week, we're going to assume that these 2 genes, the color gene and the shape gene, are on different chromosomes. So I'm going to have one chromosome here that has big A, little a, and I have another chromosome that has big R, little r on it. So when meiosis occurs, when these diploid cells become haploid cells, one of these two homologous chromosomes is going to segregate into the gamete, and one of these two is going to segregate into the gamete. So what possible combinations could I have in my gamete? Can you name one of them? So what possible combinations of these letters would end up in my gamete?
Big A, big R.
Okay, a big A and a big R. So it's exactly correct. That would be one of them because it would be one of these two and one of these two. So I'm not going to get two of these two. That would be non-disjunction. I'm not going to get two of these two. So that would be non-disjunction as well. I'm only going to get one of these and one of these, and I'm always going to get one of each of them as long as meiosis occurs correctly. So big A, big R would be one of my gametes. What else could be produced? Okay, I could also get a little a and a little r. Anything else? I could get a big A with a little r, or I could get a little a with a big R. Right? Okay, so one way to kind of use a pattern to do this is to use the FOIL method, which you've probably used in math before. So first would be the first of each of these. So this would be the F in FOIL. O would be the outside. So that one with that one would be the O. I would be the inside, which would be this little a with the big R, and L is the last, right? So I've got the last A and the last R. Okay. So first would be big A, big R. Outside would be big A, little r. Inside would be little a, big R. And last would be little a, little r. So if that helps you, that's a good method for doing this. Only works if you line them up like this. So if you mix up the A's and the R's in your parent, it's not gonna work. So these are our four gametes. Right? This one would have the same four gametes. Right? They're also going to have big A, big R, big A, little r, little a, big R, and little a, little r. So when I make my Punnett square, I now need to put these gametes on each side of a giant square. So here we go. I have big A, big R, big A, little r, little a, big R, little a, little r on one side. On the other side, I have big A, big R, big A, little r, little a, big R. Yep, that's a B. Should just use B. Little a, little r, right? So now I have a Punnett square that is 16 squares, so 4 by 4. So again, you can use that trick that I taught you on Friday, that if you have doubles of any gametes, you can cross them out, but I don't have doubles of any gametes here, so I can't cross any of them out, so I have to do this. At this point, to double check that you've done this properly, you should have gametes that have half the number of letters as your parents. Because the gametes are produced through meiosis, they should have half the number of your parents. If your parents had 4, they should have 2. And you should have gametes that have one of each of the letters. So this is again why we choose one letter for a trait, A for color, and we use different forms of that letter, big A versus little a. And you'll see when we get to multiple alleles, we'll actually use superscripts on them rather than changing the letters. I wouldn't use a Y and a G because that's going to make it confusing when I get to a dihybrid cross. I chose R for shape, so big R versus little r. Right? Okay. So I have one of each. The number one mistake I see on this is going straight from this to that and trying to use something similar to what you did in monohybrid, writing AARR on the top of it. You're not going to get the right answer that way, okay? Because A is not what the gamete looks like. big A, big R is what the gamete looks like. Okay, so now I fill this in. So something like this. So you'll notice again, something that I do that you may not have thought to do originally, I group the letters within the offspring. So I don't write big A, big R, big A, big R. I write big A, big A, big R, big R, because this will help me analyze it later on. When I have a heterozygote, a heterozygous trait, I write the capital letter on the left. And I always keep them in the same order. I'm always going to put the A's before the R's so that it's easier to analyze later on. So, all right. So what would go in this box? Big A, big A, big R, little r. Right. Okay. So I'm going to fill this in, and you can double check me to see if. All right. Lots of R's. All right. All right. So now I want to analyze it. Okay. So these are massive problems. They take a little while. So in order to analyze this, I'm not going to have you do a genotypic ratio for these. So it may say that in some of the handouts. We'll see. I don't remember where I put that or not. But I'm never going to ask you for genotypic ratio because it gets really cumbersome. And what we're really interested in is what do the offspring look like. So we're going to look at phenotypic ratio. And so the phenotypic ratio is going to be the ratio of individuals that are dominant for both traits. So it'll be yellow / round. We're going to have individuals that are dominant for color, so yellow and wrinkled. We're going to have individuals that are green and round. And we're going to have individuals that are green and wrinkled. All right. So to be yellow and round, so this is the phenotype. What genotype would give you yellow and round? All right, big A, big R would be one of them. What else would give you yellow and round? All right. And we also have big A, big R, little r. And what else? Oh, I didn't quite do that right. What else did I miss? There's one more. Big A. There we go. Homozygous for A and heterozygous for R. There we go. Okay, so those four will give you a yellow round. There's four possible genotypes giving you the same phenotype. Another way to write that possibility is it has to have a big A and a big R. What the second letter is doesn't really matter, right? So how many times do we have that? So we've got one here. This one? Yep. Here. Is that one? Nope. Nope. Yep. So how many do we have? 9. Okay, so we have 9 of these. Here we go. All right, yellow wrinkled. You have to be big A. And what do you have to have for the R's? Little r, right? So how many times do we have that? We've got one here, and one here. So 3. To be green and round, I've got to be little a's and at least one big R. Right? So I've got small a, big R, small a, big R, small a, big R, 3 of those. And then I'm left with little a, little r, and there's only one of those, right? This should add up to 12, or 16, sorry, 16, 12, 13, 14, 15, 16, yeah. So awesome, it worked, right? So that's the very systematic way of doing this, is start with identifying your traits. Do your parents, figure out your parents' gametes, put the gametes on both sides of the Punnett square, then count the phenotypes. So write out all your phenotypes and then count them. And be very careful that you don't miss any or you don't double count ones. So I put little dots in them when I'm done with them. If I'm just using a pen or a pencil and I don't have markers, I might put a triangle or a star or something like that, and I'll try to make it a little bit different so that you can count them up. Because it's very easy to make mistakes when you're doing this. So just be careful with them. And when I ask you questions like this on like a test, the reason I like to have you write it all out is that I can see, oh, you just missed one little spot there, but every answer you had after that was based on that one mistake, and I can give you partial credits on things. So, so what I'm looking for is that. Okay, questions on this? We're going to do some practice problems, but so these are a little more complicated. These are as complicated as it gets, as we'll see that anything more than 2 traits, you stop doing Punnett squares unless you're doing it as an activity for a class or something like that. Geneticists do math rather than Punnett squares. It becomes a little bit easier, right? Okay, so let's. I'm just going to talk a little bit more about this, and then we'll do another practice problem. Get this back. Okay, so here, this graphic kind of did the same thing. It actually took you from parents that were homozygous for both to give you all big R, big G in this case. And then it did a dihybrid cross, and it shows you the offspring. And here's the phenotypic ratio of 9:3 to 3:1. Again, this is all based on what's happening in these cells during meiosis. So a heterozygous individual, heterozygous for 2 traits, during meiosis, the homologous chromosomes are going to separate in meiosis 1 so that in this case, a big R went with a big little g, and in this case, a little r went with a big G. And meiosis 2, you end up with gametes. But we have independent assortment, so it could have happened like this, or it could have happened like the one on the top. So you have alternate forms. So you've got 4 possibilities, and it's just as likely for any of these possibilities to be the sperm or the egg that is fertilized because it's random fertilization. So independent assortment leads to what we're seeing in dihybrid crosses. Remember, as of now, we're assuming that all these traits are on different chromosomes. There's going to be something different that happens if we have traits on the same chromosome, and we'll talk about that next week. Luckily for Mendel, all the traits that he looked at were either on different chromosomes or they were really, really far apart on the same chromosome, and we'll see why that basically acts like it's on different chromosomes. So we'll look at that in a bit. Okay, here's a question, and let's see how we can. All right, see if you can answer this one. Yep. Yep, exactly. Good, good. All right, we're gradually narrowing in on it, it looks like. So I only have a few more minutes, so I want to do this with you as quickly. So the next step is figuring out your parents, and a heterozygous red, heterozygous wavy. Heterozygous for red is going to be big R, little r. Heterozygous for wavy, in my case, is big H, little h, crossed with a blonde straight, and this is where a lot of you went off. A lot of you just did that cross with itself, which is like what we did in the last problem. It's a different problem, different second parent. To be blonde-haired, what does your genotype have to be? 2 little r's. And to have straight hair, you have to be little h, right? So that's your second parent. So if I do FOIL on this, I get big R, big H, big R, little h, little r, big H, little r, little h here. If I do FOIL on this parent, they're all little r, big H, right? And here's where you can use that shortcut. All of these are the same, so you can cross out all the duplicates and give yourself just one. This will make your Punnett square much, much easier to make. Or you can do all of them if you want. But for the sake of simplicity today, I'll just do this. So I'm going to take one parent, put it on the top, big R, big H, big R, little h, little r, big H, little r, little h, and the other parent on the side. I end up with this, which each of these has a different phenotype. This would be red wavy, this would be red straight, this would be blonde wavy, this would be blonde straight. So it's 1 to 1 to 1 to 1 of all of those. You would have gotten 4 to 4 to 4 to 4, but it's the same as 1 to 1 to 1 to 1. So the answer is C here. All right. So practice problems like this. We'll do more problems like this next week. We'll have other problems to try in class that use this technique. But definitely do the worksheet on your own. Do the homework problems. I will post answers to the worksheet questions at some point, and you can practice doing those. So, all right.